Hello everyone. In today's lecture we're going to talk about gene mutations. So in our previous discussion we learned all about how DNA sequences are used to create mRNA copies, the process of transcription, and then those pieces of mRNA are then translated at the ribosomes using again those three-letter codons to create a series of amino acids. Eventually, that chain of amino acids will go on to be folded into a protein, right? So we talked about primary, secondary, and tertiary, and even quaternary structure of proteins, how those things can be looped and kind of clumped and made into the shape that they need to be in order for the protein to have that function. But what happens if there's a mistake? Mistakes happen. So when we talked about DNA replication, we were talking about how the process occurs at multiple replication forks on your chromosomes, and it's happening, fairly quickly, right? So we're talking about 100 bases or so every second, and so at that kind of speed, you're bound to make mistakes. So what happens when we have mistakes in the copying process of the genetic code? We talk about those, and we're going to refer to those as mutations today. So we're going to talk about different types of mutations, and we're also going to talk about the causes of those mutations. So first of all, a mutation is very simply a change in a gene's nucleotide base sequence. Okay, so again, we're being very specific about, we're talking about the coding regions of our DNA, not the non-coding regions of our DNA, right? So mutations occur when we have a gene that codes for a protein and there's some kind of change in the nucleotide base sequence. Now, from the perspective of how life has evolved on this planet over millions and millions of years, mutations are great. Mutations are important because they have enabled us to produce mutations has enabled life to produce individuals who have different variations of traits. And sometimes those variations are more successful in whatever environment they're in. And when they become more successful and they do better when it comes to surviving life, finding food, water, shelter, space, and a mate, and if you have traits that make you better at that, those traits are more likely to be passed on. So to be sure, mutations are an important part of life's evolutionary history. It's not necessarily always just bad and leads to disease or disorders or complications. So a mutation is referred to a sequence that is present in less than 1% of a population. It can occur at the DNA or chromosome level. This particular lesson, we're focusing on the DNA level. If we have a change that becomes more widespread, it becomes a polymorphism. So again, if it's great, it becomes present in more than 1% of the population. So again, if something occurred and started small in a small group of the population, but because of natural selection, which we haven't gotten to yet, we'll get to later this semester, but when we get to natural selection, we find out that that particular allele combination, the combination of traits, is really, really successful, you may see a mutation become a polymorphism over time. And again, the effect of mutations can vary, right? So we can have, these what are called loss of function mutations. Something happens and that protein doesn't work the way it's supposed to anymore, and therefore we're losing a function. These tend to be recessive types of gene traits, if you will. We can also have gain of function mutations. So you're able to do something that you previously weren't able to do. That tends to be a more dominant trait versus a recessive trait. And again, we'll go back to dominant and recessive. Actually, I think we start that in our next lecture. We'll start talking about traits as being either recessive or dominant, depending on how they're inherited. All right, so the term mutant, it refers to a phenotype, right? It usually connotes something that is, abnormal or unusual or uncommon. But again, this is an important part of living things, right? To have these different types of mutations that potentially can enable us to better survive or better adapt to changing conditions. So Our evolutionary relatedness to other species allows us to study mutations in non-human species. So we can actually sort of figure out, as I mentioned in the very first lecture, I think we sort of have this molecular clock based on how many mutations have occurred between us and other species. We can actually use that as a way to determine how closely related we are to other things. All right, so let's move into types of mutations. So the first one we're going to talk about is a very simple mutation referred to as a point mutation. In this case, we are just substituting one DNA base for another. So in the image here that I have, so you can see the original base triplet in a DNA strand. Okay, so I've got both strands here. Now let's say that that process is replicated. So this little fragment was replicated, but a mistake happened. And instead of the T nucleotide, which is supposed to go here, a C nucleotide went here instead. Okay, so this is a mistake. All right. So what happens is we have, when replication happens again, we will have one copy that will be unmutated, that will have the original sequence. And because remember, both sides serve as a template, but the other side is going to have that genetic mutation, right? And then from this point on, every time this copy of DNA is copied, no mutation. And every time this particular one is copied, their mutation will be consistent. It will be repeated over and over and over again. So we can define point mutations as either being transition or transversion. If a purine replaces a purine or a pyrimidine replaces a pyrimidine. If it has a purine replaces one or the other, or vice versa, we call it transversion. Don't worry too much about that. I'm not expecting you to know transition point mutations versus transversion point mutations. It's just sort of for your own information. So what can happen when we have a point mutation? So we can have something that becomes what's called a missense mutation. So this changes one amino acid into another. So let's say this was our original set of codons that we had in our mRNA sequence. In our DNA sequence, which would then be copied into mRNA and then becomes these amino acids. We have methionine, glutamine, alanine, and glycine, right? So if we change that A and it becomes now a C, we now have a different amino acid, okay, aspartame in this case. That's a missense mutation. We have one amino acid changing into another. We can also have what's referred to as a nonsense mutation. This is when it changes one amino acid into a stop codon. So again, a nonsense mutation means you completely stop the production of amino acids right there. So no more amino acids are actually linked onto that chain because we hit our stop codon. All right. So again, point mutation can be a missense, can be a nonsense. Other things we see when it comes to mutations, we can also see frame shift mutations, right? So a frame shift mutation is when we actually remove or add a whole base. Okay, so again, let's say that when we copied this sequence, we forgot to copy the G. It just didn't happen. It disappeared. And so now the reading frame has completely shifted over. So now again, we go back to our original strand here, which was methionine, glutamine, alanine, and glycine. And now we have every amino acid down the line is now impacted by this shift. So rather than just changing and swapping out one amino acid, we are now swapping out all of the amino acids further down this chain. The one caveat here is that if you remove 3 bases or you add 3 bases at a time, you don't alter the entire amino acid sequence. You would just have one amino acid sequence, one amino acid that is different, right? So either it's missing that amino acid or you added a new one to the sequence itself. Because again, remember, we read these in 3 letters, right? Each codon is 3 letters. So any multiple of 3 won't shift the reading frame. It just might add or delete amino acids. Okay. We also have a situation where you can have a duplication when you have a whole part of a gene sequence repeated. We see this more in when we start talking about chromosome abnormalities, which we'll hit on a little bit later. This is something that we see kind of a little bit more that you can see with entire pieces of chromosome that sometimes this happens to. So this is kind of a nice mutation analogy. So I like this because it sort of really kind of makes clear what the consequence of these different mutations happen to be. So if this is our normal DNA sequence and we say it's a sentence, these are all 3 letters long, right? So the one big fly had one red eye. A missense point mutation might change a single letter. Okay, so we have a single letter. THQ doesn't make sense, but the rest of the sentence does. A nonsense point mutation stops the sentence altogether. A frame shift mutation inserts a letter where it doesn't belong. In this case, we inserted the letter Q, and it changed all of the other words in the sentence. Excuse me, which again is like the amino acid sequence. A deletion mutation is if we're missing a word. Insertion would be if we have a brand new word, and duplication is if we did something twice. Okay, so again, kind of a nice analogy for you to sort of reference when you're thinking about how mutations can alter amino acid sequences. There's also a difference between where these mutations can occur. If a mutation occurs in the germlines, so remember we're talking about spermatogenesis and oogenesis, and we were talking about the formations of eggs and sperm. If the mutation happens in the process of forming gametes during meiosis, then the gamete itself and all of the cells that are created are going to have that particular mutation, right? So the mutation wouldn't necessarily impact the person themselves or their cells as far as their outward phenotype goes, but their offspring would be affected by a germline mutation. Somatic mutations will occur just during meiosis, and only certain cells are impacted, right? It's not a whole organism thing. It is just whatever part of the body it happens to happen in. So for example, cancers, right? This is a really great example of what will happen when we talked about control of the cell cycle. We said there were multiple genes that were responsible for those checkpoints that prevented a cell from continuously replicating. What happens in the case of cancers is that we have your DNA becomes fragile for various reasons, and we'll talk about mutagens in just a moment. Mistakes are made in the copying of those particular cells, and then you might have the checkpoint genes are impacted. And when they are, it can lead to continuously replicating cells. So speaking of mutagens, how do mutations happen? So there's one of 2 ways that you can see mutations happen. They can be purely spontaneous. And again, a spontaneous mutation is just one of those errors that happens because we're copying DNA really, really fast, and sometimes we make a mistake. Or you can be exposed to some type of a chemical or radiation, and any physical agent that causes a mutation is referred to as a mutagen. So here, spontaneous mutations, again, results from errors in DNA replication. So whoops, we made a mistake, we're going too fast, the proofreading mechanism didn't work here, and so we have a mismatched base pair set, right? So the rate differs between genes, but the spontaneous mutation rate is about, there's a 1 in 100,000 chance of mutating. Every time, again, you copy, say, 100,000 base pairs, very likely one of those is going to probably mutate. So the larger the gene, the higher the mutation rates, right? And so you are a walking person that has multiple new mutations, right? So there have most definitely been mutations that have occurred in you since you were born, right? Because as we're copying all these cells and we're growing and we're changing, things happen and mistakes happen. And so the spontaneous mutation rate means that you have a bunch of new mutations. Now, whether or not they mean anything is entirely different, right? Because even though we can see, like we talked about some of those consequences, the frame shift and the point shift and all of those things, if you remember in our last lecture, we talked about the redundancy of the genetic code. So in some cases, you can make a mistake and nothing happens because there are 3 or 4 different codons that will code for the exact same amino acid. So sometimes those changes don't really matter and they don't make a difference. There are places that are called mutational hotspots. There are places where we have repetitive sequences or palindromes, where we have the same sequence forwards and backwards. It's really likely to make mistakes here, right? So if you think about just in your own typing and you're sort of typing on your computer, there are certain words that you're more likely to misspell. And so same thing with mutational hotspots. It's just really hard to make perfect copies of these areas, and so we're likely to make mistakes. So then if mutations aren't spontaneous, then they were caused by a mutagen. And there are different ways that you could be exposed to mutagens. And I say you, I mean living things in general. In some cases, we actually do this on purpose. So we will induce mutations in different things in our labs, like fruit flies or mice, for example, in order to see what that gene does and study what that gene does so we can understand it better. Oftentimes you will be accidentally exposed. This could happen in your workplace. It could happen in some type of an industrial accident, different types of medical treatments, radiation. Any type of radiation is a known mutagen. So if you are at all exposed on a consistent basis to radiation, hopefully your workplace is required to provide you with proper protection from that radiation, because we do know that any type of radiation happens to be a mutagen. And then you are also just exposed, just being out in the world, stuff coming from space and sunlight. And there are even radioactive minerals in the earth, things like radon, are forms of natural exposure. And there are things we can do to protect ourselves in certain cases, but we can't completely prevent exposure from these things. So again, nuclear power plants, for the most part, you are perfectly safe if you are near a nuclear power plant. However, if there happened to be some tragic incident and we had a core reactor meltdown, there is radiation that is there on site of the reactor. It potentially could leak into the community and be a problem. But again, that's very, 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 very rare. So nothing to worry about. So what happens? Okay, so we have mutations. They're either spontaneous or they're caused intentionally, or they're caused by some sort of a substance like a mutagen. What can be the consequences of them? Well, the first thing that we're worried about is we're worried about harmful mutations. These are disorders and diseases that are caused by proteins that are not functioning properly, right? So when we have a disease where we have a cystic fibrosis, for example. Cystic fibrosis is caused by a mutation in a gene that is responsible for essentially keeping the mucus that our body produces nice and thin and liquid so that we can clear it really well. And in folks with cystic fibrosis, they have a mutation that actually causes that mucus to be really, really thick. And it's difficult to clear from the body. And so the biggest threat for those folks is that they are more likely to catch different infections, and they're harder to fight when you have cystic fibrosis. Other mutations can be beneficial. We don't notice these very often, but these are the types of mutations that often lead to evolution, because these are things that can enable people to better survive and better to able to potentially pass those traits on to their offspring. So there is a change in a gene that prevents HIV infection. So folks who have this are immune to HIV because their cells are protected from it. And then more likely than not, no effect whatsoever, completely neutral. Because again, you have the redundancy in the genetic code. And because of that redundancy, you can have multiple amino acids, multiple codons that code for the same amino acid. So a change might mean nothing at all. So just some examples of beneficial mutations. If you are able to digest lactose, that is a mutation. So we as humans are unique in that we are a mammalian species that is able to tolerate lactose beyond infancy. All other mammals can only drink their mother's milk for so long, and then as they age, they lose their lactose tolerance. The enzyme lactase that digests it is no longer produced. And that's so that they can't feed off of their mothers anymore. They have to eat regular food. This mutation probably occurred multiple times in human history, but probably right around the time where we started to have a close relationship with herding sheep and goats. They became a great source of protein in that you didn't have to kill your sheep or your goat. You could actually just use the milk from it then, and it became a really important source of protein. Again, I talked about immunity to HIV, resistance to atherosclerosis. So there are some folks whose arteries are able to sort of clear that plaque a lot better than others. So that's fantastic for reducing your risk of heart disease. And then depending on how you feel about twins, hyperovulation actually is considered, again, potentially a beneficial mutation in the fact that instead of just having one child, we can do two. So that's it for this lecture. So pretty short and straight to the point. So again, you should know the difference between somatic mutations and germline mutations. You should know the difference between point and frameshift mutations, what mutagens are, and how they're caused, and what are the possible outcomes.